Ja, Nick ist im Haus. Heute geht es um Biotech, nämlich Oxford Nanopore, glaube ich, seit dem Start oder auf jeden Fall seit langer Zeit eine Position in unseren Fonds. Und wir werden jetzt mit dem Unternehmen sprechen. Nick geht da natürlich in die Details mit seinem Wissen in das Produkt rein. Und ja, da das Unternehmen in England, UK sitzt, werden wir jetzt gleich ins Englische wechseln. Und ich würde sagen, auf geht's zu Oxford Nanopore. Rosemary, thank you very much for joining us today. So maybe we start just a little bit about yourself, your background, who you are, and what Oxford Nanopore is doing.
Sure. Thanks, Frank. Thanks, Nick. So my name's Rosemary. I've been at Oxford Nanopore for approaching 10 years now. I came to Oxford Nanopore through a career in life science distribution. So I was at a company that made or that sold over 1 million products into labs all over the world. And 10 years ago, I saw this little thing called a MinION, and it completely and absolutely blew me away. So when I had the opportunity to join Oxford Nanopore, it was like all the Christmases came at once. At Oxford Nanopore, my role, I look after all of our product and program management functions and also our marketing teams. So my role at Nanopore is to live and breathe with our innovation groups, understand what they're doing, what they're working on, what our technology is capable of, and then taking any new innovation through our production, supply chain teams, our commercial teams, and effectively work on the commercialization of our innovation group. It's something that is never still. There's never a dull moment. Our innovation team are capable of coming up with an enormous amount of either new products, but also a lot of improvements to our current platform. So it's every day. Every day is a dream, and you come to work and there's something new to talk about, think about, and more importantly, consider in terms of how we release it out into the customer world and what it can do, what it can do for our customers.
That's great. Before we dive deeper into the product later with Nick, so maybe can you try to explain for kind of normal people, what are you doing? What's so revolutionary? What's your vision? What do you want to bring as a revolutionary tech to the world?
Absolutely. So DNA sequencing is, or the discovery of DNA is something that humanity has been working on for over 100 years now. It took us about 100 years from discovery to sequencing the first ever strand of DNA. And that was on very, very basic instrumentation. And what we've done since then, what scientists have done since then is break through that. So go from sequencing a really small genome to sequencing the human genome. And the first draft assembly of the human genome took 10 years, billions of dollars, and took a multitude of labs around the world, hundreds of people. And that was something that was completed in the early 2000s. From the early 2000s until around the sort of 2014, 15, 16 time, what the technology, what the market was doing was really scaling this capability. So how do you go from one human genome costing billions of dollars to sequence to human genomes costing under $1,000 to sequence and being capable of many being generated worldwide or in a single lab. So what the DNA sequencing industry has managed over these last 100 years is take something that wasn't possible, become possible, and scale it. And then enter Oxford Nanopore in 2014 with our first product with MinION. And the reason that we're really looking at disrupting this market is because today, to sequence DNA, you need to invest in a really large piece of equipment. It needs to go into a centralized facility, a big lab. You need to have expert hands running it, and that's fantastic. You know, it's really delivered an enormous amount of innovation throughout the years. However, there's a lot of value in DNA sequencing, either sort of human, plant, animal, bacterial, and we believe that a lot of that value is actually laying by being able to bring the sequencer closer to the sample. And so our technologies are handheld technologies. So you have the next generation of MinION here. And so this is really why we think we're disrupting this market, because we're taking very, very large instrumentation that is currently required to sequence DNA today, and we're making it portable and making it accessible and easy to use. In terms of the type of data that we can generate as well, that's also a step change. So if you can think about what the standard way of sequencing today, which is what people call SBS or short read sequencing, you can consider it almost like taking a black and white photo of the DNA. There's bits of information missing. So there's no methylation information, there's no epigenetics in that photograph. And there are large variants, genetic variants that are really important in a lot of, be it rare disease or cancer or other complex genetic conditions. Things called structural variants that are just not available in today's technology. So not only are we disrupting the market by bringing out new form factors, smaller form factors, but we're also generating a different type of information. So we're very information rich in terms of the DNA that we sequence. And that information is proving to be incredibly valuable to researchers around the world, be it in human genetics, plant, animal, or bacterial.
If I'm not mistaken, you have been part of the latest Apple keynote, where they had the M3 shown as a very capable processor. What exactly did they show in the keynote? So in kind of easy terms for our listeners to understand this.
Absolutely. So when we started our sequencing journey, our MinION, the little handheld that I just showed you, that device could sequence maybe 1 gigabase, 1 billion bases of DNA in 48 hours. So you could plug it into a standard laptop, and that was okay. The standard laptop could process the data. What's happened over the last 10 years with our technology is we've now increased our ability to sequence about 50-fold. So you can now get 50 billion bases from a small handheld sequencer. And also the algorithms that we use, we've borrowed a lot from the machine learning algorithms that are used every day in technologies such as Siri, so voice recognition technologies. And those algorithms have meant that our accuracy is now well over 99%. And what we found was that the standard laptops would just not keep up with that at all. So we've been working an enormous amount with the GPU technology makers on the planet. So our larger instruments we've got an amazing collaboration with NVIDIA. But really for us, MinION is a product that should be capable of being used by anyone. Apple has the market-leading user interface and user experience in terms of compute. And so when Apple started to come out with their new silicon chips and the M1s, the M2s, and now the M3s, we got really, really excited because it meant that finally we had a very, very simple-to-use compute platform in Apple, pairing up with a really simple-to-use sequencing technology so that we can just take MinION into a whole new space. So really, it's what we've been waiting for the last sort of five or 6 years, is for the compute industry to catch up with what Nanopore have been doing in terms of data generation.
Perfect. So Oxford Nanopore enables DNA sequencing not with big machines and external partners, but you can really bring it to where you are, whether you want to analyze this from a human body or from other parts where DNA sequencing brings value. And then with the latest chips from Apple or NVIDIA, you basically can also analyze it everywhere because it's available technology. So perfect. So Nick, maybe we dive a little bit deeper into the nitty-gritty details, the interesting details about the mechanics and the biotech behind it.
Sure, Frank. So maybe for our listeners and viewers, it would be interesting to maybe get a little bit more understanding of the technological difference in terms of the sequencing. I think a good example is always that what other companies are doing is taking snapshots, photos, copies of a DNA fragment, and using different light signals basically to determine the sequence technology or the sequence, the DNA sequence. But how does Oxford Nanopore is different in terms of the technology in itself? We learned that it's using smaller instruments, have it everywhere for everyone to analyze it. But there's also, in our perspective, even more distribution that we're seeing here.
Yeah, absolutely. So the easiest way to think about it is that the current technologies on the market are looking at DNA. So they're taking photographs. It's light based measurement, and the way you do that is you add a fluorescent chemistry to each DNA base, and it lights up a different color, like the colors on your Christmas tree, right? As we're getting towards that time of year, and that that's been the fundamental technology that we've been working off for the last couple of decades. The way that Oxford Nanopore works, it's an electronic sensing, so it's not light sensing; it's electronic sensing. So we have very small nanopores inside our flow cells, inside our consumables. Nanopores actually exist in nature. They're inside all of our bodies. They are the little channels that allow two cells that are next to each other to talk to one another. So what happens is sort of pieces of information flow through these channels between cells. And what we've done is we've taken that concept, we've taken those nanopores, we've engineered them, and we've put them inside our system. So what we have is a donut shape, a nanopore shaped, and the DNA sequence, the whole entire strand, not little pieces of it, but the whole strand of DNA is pulled through the nanopore. And as it goes through the nanopore, we feel it. A little bit like, Braille, if you imagine if you're a blind person, you're feeling what you're reading. It's the same with nanopore. We actually feel the native DNA. So the benefit of that is that you're not looking at a fluorescent reaction, because that is a sort of a pseudo result. It's a chemical result. You're actually touching the DNA and sequencing the DNA itself with our technology. And that is fundamentally why our instruments can be completely scaled, from anything that is handheld to the larger pieces of equipment that we have, like PromethION, that do live in the large projects, in the large laboratories. It's because we don't need lights, we don't need enormous cameras and really massively advanced camera systems. We don't need that. All we need is an electronic sensor. And the main thing that Nanopore do is the consumable that goes inside, the electronic sensor is actually on the consumable. So this is also why our equipment is sort of very, very scalable and low cost to manufacture, because it's all of the electronics, all of the smarts, all of the nanopores are in the consumable cartridge that customers buy, run their sequencing experiment, and then they return the cartridge to us and buy another one.
Yeah, fantastic. I mean, we've been talking a lot about DNA sequencing now, because this was basically the first thing you did. But in terms of the technology, because it's only electricity, and as long as we have some kind of charge in our sample, we can basically use the nanopore technology. And I think we're actually seeing like technology developing in this area, and you already have RNA sequence in it. But what do you think can be analyzed with nanopore sequencing beyond the nucleotide sequence?
Yeah, that's one of the really interesting things about our platform, is that you, just by changing out the nanopore, which is something that we can do very quickly, you can change from reading DNA to reading RNA to reading proteins, reading small molecules. So what we've done is we've created an underlying platform, which is our hardware, our consumables, our software, our reagents. But we can just change certain components of those, and then you have, for instance, a protein sequencer, or change other components and then have something that would sequence, for example, sugars, or other small molecules. So that's one of the really exciting things that we see for ourselves in the future, is moving from beyond being, recognized as a platform that brings a lot of value to DNA and RNA sequencing, but then taking that platform into completely new spaces where there are just no technologies today that can deliver the information that we think we can deliver with protein or small molecule sensing.
Yeah. When we analyzed the Oxford Nanopore, and also for me at my time as a researcher, I was always struck by the community of researchers around Oxford Nanopore, and I think this is something special in a sense, and was really early on. Can you a little bit tell us more of the thoughts behind it, or the rationale why to include researchers so early, and how this developed, and what you think is the benefit or the driver of this community?
Absolutely. Well, the community for us is completely central to the way that we commercialize our product. We often get asked, do we compare ourselves more to a life science tools development company or a tech company? And I think we kind of behave like a tech company in a life science tools market, so a little bit of both. But if you kind of rewind the clocks back to 2012, when we first showed MinION at AGBT, there was quite a lot of doubt in the sequencing community whether this technology was even going to work, whether it's going to be possible, or there was a lot of thoughts to say, well, you might make a sequencer that works in one place, but I bet that if you shipped it around the world, it wouldn't work. And so the community came out of that absolute necessity to, with our first product launch, show that this technology does work. And so we started the MinION Access program. We allowed researchers to submit their proposals, what they wanted to do. They could then get a MinION for only $1,000 deposit. And we made sure that we had researchers in the US, in Australia, in Europe, in Japan, in all corners of the world. And we started shipping. And we needed a community for two things, really. First of all, to make sure that that the technology did work, and we could dispel all of the sort of apprehensions that were out there in the market about whether this technology was real or not. But the other thing as well was the data was so different, right? The type of data that we produce is completely different. And so the community, it was really important to harness a community spirit where you had really very, very talented algorithm developers, really talented informaticians who were starting to look at a completely new type of data and try and make sense of it and try and create tools for it. And that was the kind of the sort of the reason that we really went out from what we would call the bottom up approach. Make the technology available to everyone, let everyone have a go, and then for us it was instrumental, because when you have hundreds of people giving you feedback, it completely shapes the way that you're running your R&D teams, what they're working on, what are the critical things that people think, and it's really useful to have that from hundreds of people instead of, say, 5 or 6 in a few controlled labs. So yeah, it was a big gamble, but hopefully one that worked in the long run.
I mean, I guess it already paid out quite nicely because of the amount of tools that are developed and the amount of publications that are now coming out every day, basically showing different applications. And I think it accelerates the development and the use cases of this technology. And I think this was, at least for us, a quite nice move, and we really appreciate this. So maybe moving to the application and the markets you're currently serving, what is the main application you're offering? It's DNA sequencing in a sense, right?
Yeah, well, that's always a tricky one, right? One of the beauties about letting a technology out that everyone can afford is that you then end up with 700 different use cases. But what we try and do is we try and sort of coalesce them into key focus areas for us. So DNA and RNA sequencing are the lead application of our technology, and our focus areas, because it's what we hear our customers speak about the most, is around whole human genome sequencing, and that can have a few flavors. So that can have the flavor of sort of driving these telomere to telomere assemblies, looking at rare and undiagnosed disease. There's, of course, the massive space of oncology, where methylation, epigenetics, a lot of the capabilities that we bring to the market, including speed, are enormous wins. And then we have our pathogen arm. So Nanopore, one of the first big applications, of course, for it was sequencing Ebola in Sierra Leone in 2015. And of course, it's since then been used for outbreaks like Zika and, of course, COVID. So we try and sort of bring all of these applications together. There's also other things like single cell. There's de novo assembly of plants and animals, and all of those other additional things. But the three real focus areas for us are the human genome sequencing, oncology, and the pathogens.
Yeah. And the current customers are mainly like R&D. We see a shift towards clinicians, and this is obviously a huge market for various reasons. But going forward, what do you see like potential new customer or segments? What do you see like Oxford Nanopore going towards, and something that the competitors maybe are not able to?
Yeah, so it's really kind of the impact that the technology has had in the research labs. What they've shown that the technology can do is getting a lot of applied markets very, very excited. One of the big ones, of course, is clinical. So we are investing really heavily in getting our platform or parts of the platform locked down and frozen so that they can be deployed in clinical settings. And they're the kind of, again, the benefits that we have there. One of the absolute major ones is the time to results, and that's not only because we can sequence exceptionally quickly, we can generate data very quickly, but you don't need to batch samples on our system in order to get the price per sample that you need to return a result. So, for instance, on other technologies, if you're a cancer patient, the other technologies might need to wait for 96 people to show up in order to fill an instrument for the instrument to run. And so it's not necessarily that the instrument runs slower from other competitors. The problem is that you have to wait for 96 people to show up with that condition before anyone even switches the machine on. Whereas with Nanopore, because our sequencers are completely scalable, one person can come in and we can generate the library, sequence it, and then when the next person arrives, you can do that as well. So we're taking so that's a really big pull point from the clinical application perspective as to why people want to use Nanopore in those settings. So we think there's going to be an enormous amount of new customers in that space. We're also incredibly active in things like biopharma, ag bio. There's a lot of other industries where access to real-time DNA or RNA information are a big win. And then, of course, we still have the research market, which for us is critical, because as we launch new capabilities like protein sequencing or small molecule sensing, right, everything starts with the research platform. So you have for us our research platforms give us our our sounding board for our technology, give us the proof of principles, give us those early concepts of what can be done with the technology. And then we work with our customers and with partners in terms of when it comes to commercializing those particular applications, how do we best serve that next market along, be it a clinical market or a food market or a biopharma market. There are different experts in all of those spaces.
When we talk about Wright's law, so that when you scale, things get cheaper, you look at the battery inside an EV where the cost really drop sharply. When you go into scale, like how much scale possibilities do you have to reduce the cost? What kind of scale could Oxford Nanopore grow in? So how do you see cost and scale?
Yeah, so we've got a really active R&D pipeline. So the MinION flow cell that I showed just a second ago, that is generating sort of 50 billion bases at its maximum. We have the PromethION as well, which is our lead platform for human genetics. So again, you've got a different sized flow cell that's got. So this flow cell has almost 3,000 nanopore channels that are active at any one point. And on this flow cell, you can sequence a human genome, for instance. We have a lot of different types of flow cells in the pipeline, and we can do a couple of things. So on this flow cell that we have now today, on the instrumentation that we have today, we can keep upgrading the chemistry, so that you go from one human genome to 2 human genomes to 3 genomes to 4 genomes. And we have all of those proof of concepts in-house, and we have faster motors, we have different chemistries that can really drive the output. And that means that anyone who's bought a device, a PromethION device, is going to go from 1 genome per flow cell to 2 or 3 or 4, and therefore get to a price of about $200 a genome on the current product that we sell today, without having to buy a new box every time you make a chemistry change. Beyond $200 per genome, we have our future roadmaps where we have capabilities where instead of having 3,000, you could have 10,000 or 100,000 nanopores on a flow cell, and that will really sort of pave the way to much lower and much faster human genomes in the future. So we have the technology that we have available today, capable of with chemistry upgrades, capable of taking us to a market-leading position for the market that we currently occupy. And then we have future pipelines that take us way beyond that. So it is, again, very, very scalable. I think we've shared that at our Capital Markets Day, that our innovation team don't stop thinking about the future. And that's part of the excitement. That's part of the appeal when it comes to our technology, is that it's never standing still. And but in order for it to move, it can be very simple changes that we can do to an existing product today that will take us to the next level of performance.
Makes sense.
I mean, the most simple change we recently saw was basically a light shield. Improving the output amount. So what would your suggestions be of, do you see more improvements on a chemistry nanopore development side, or do you think that the software development and certain applications are potentially a driver to increase output or accuracy in a sense?
So we work on those sort of three. We work on 3 pillars in our R&D groups. We work on accuracy, output, and reproducibility, reliability. Those are sort of the 3 pillars, and it actually takes every piece of the puzzle to work together in order to achieve it. So if we have potentially a new nanopore, then the software team need to tweak all the software in order for that new nanopore to be in good shape, and a new algorithm needs to come in. So actually, all of the teams are constantly working together. But we do. So there's a lot of opportunity in the proteins that we use our protein engineering. There's a lot of opportunity there. There's a lot of opportunity in the chemistry that we run, so the buffers. There's constant tweaking of our software that actually operates the machines and our algorithms. So we will continue to see output and accuracy improvements from all of those areas, and that's why we hold dearly. We hold things like events like London Calling and our Nanopore community meetings that we have. We hold those really dearly because it's where we communicate to our customers where we think the technology will be in 12 months' time. So we constantly make sure we're bringing them along with us on our journey.
Yeah, so I mean, those improvements in terms of chemistry and the pore would also enable to bring down the cost or the throughput of smaller sample sizes or smaller instruments like the MinION or the Flonge cell. And would you eventually, or is this a vision to have an end customer product basically available? I mean, with the iPad integration, you can have it at a doctor's office basically. But I mean, it could also be intentionally to have it something like a COVID swab test, to have, if you're sick and you need a quick diagnosis, you basically sequence at home if it comes at a certain cost. Do you think this would be like potentially available at some point?
That's very much the long-term vision. And if you think about what we're doing today, we announce a lot of large projects where we're working with different countries on understanding different parts of the human genome. And so that's why we have our really large PromethIONs, the P4024, the P48. Those things can go through 5,000 genomes a year or more. And we need that in order to start understanding what is it in genomes that impacts human health. And so you create that understanding. And at the same time, we then create more portable versions of those instruments, like the P2 Solar that we launched this year. And we create those so that they can be point of care. And then we have future things like TraxION and Turbot, which also take that one step further. So very much the vision is that it will one day end up in the doctor's office. You have to go through a journey in order to get there. The first journey is understanding the science, which is what we're doing today. Then we need to get all of our products clinically approved and really well run in the clinical market. And we've got fantastic collaborations there with people like bioMérieux and the Mayo Clinic and others that we've spoken about to really drive the platform and get it into the clinical space. And then going consumer is a next step. But the thing that excites us about, and the thing that excites our customers is about our platform, is that it is the platform that can do it because of its format.
I mean, the first time we have this data in a broader range, because as you just mentioned, we're coming basically from billions of dollars not possible into this very steep curve of cost decline, and so we don't have that much data today. So you basically will get more data about what can be done with your technology every day. And then, of course, we have AI can look into this and then find more ways of saying, hey, you should look at a doctor, or this is something first stage of cancer or whatever, what you then can see while getting more and more data. So it's kind of a flywheel that's just getting started.
So you just mentioned the collaboration with the Mayo Clinic or bioMérieux, and we've seen a lot of new collaborations in the last months, let's say. And to us, this was like a shift in strategy somewhat since the IPO. It was basically setting up office, let's say, and now we see more and more collaboration. Can you tell us a little bit more about the strategic shift that we're currently seeing at Oxford Nanopore?
Yeah, I'm not sure it's a strategic shift. More, we've reached that time point where this inevitably was going to happen. So at Oxford Nanopore, we are experts in creating sequencers and driving our technology forward. When you think about, so one of the first products that we're really excited about is our TB, potentially a TB test that we're working on with bioMérieux, for example, to launch. And bioMérieux is incredibly well respected, established in all of the countries where TB is causing a great amount of pain. And so, of course, it would make a lot more sense for us to partner with someone like bioMérieux who know the landscape in order for them to make our technology more and more available there. In the same way that when we go into biopharma, there are going to be different companies that are really well established there. So the application of the product, the application of our technology is so diverse that our sentiment really is that we're better partnering with experts in different sectors for them to be able to absolutely magnify and amplify what our technology is capable of. So yeah, that's why we're building the partnership program that we spoke about at the Capital Markets Day. We always saw it sort of very early on, like, Apple in the App Store. You have different people who actually know what apps to build or know how to push those out. So yeah, there's no real shift in strategy. It's just we've actually gotten to that point that we weren't at 10 years ago, but we're very much getting there now, and we're really excited about it.
I mean, you said that you mentioned the App Store as an example of what is like as an example for something else in collaboration. And do you think that Oxford Nanopore is a point where you can actually have this App Store moment where people come in building software technology on top of your technology to enable somewhat of apps, also to say that you basically have the platform and now people can come in and extend the possibilities?
Yeah, actually, we launched it at ASHG. We showed it. We gave a demonstration of so we have an epitome platform for analysis. So when the data gets generated by the instrument, it can go into epitome to get analyzed. And what we can do in epitome is, if you have a human genome, we can say what genetic variants are there, for example. What we don't do is we don't say which genetic variants are linked to a particular disease. That's interpretation. And there are fantastic interpretation software companies out there doing a fabulous job already. So what we showed at ASHG was a link where someone would upload their data into Epitome, they would run the human variation workflow, and a pop-up window appears and says, okay, well, which partner would you like to do interpretation? And therefore, which partner would you like to choose in order to take that next step? So yeah, we're absolutely gearing towards this ecosystem where we will take the genetic information so far, and then let the user select what they want to do next with it in collaboration with these amazing companies. And that's all really kind of centered around the user experience. It's much easier for a user to run. For sequencing, sequencing should just become second nature. You do some prep, you load a sequencer, and then you're actually just thinking about your result. People should actually forget that they're sequencing, right? They just want to go sample in, answer out. And so really making sure that we've got a really robust infrastructure underneath and a robust pipeline end to end means that we can try and get customers to forget that they're sequencing, but just think about the sample they put in and the answer they got out. That's what we're really working towards.
Wow, very impressive. So we like your technology, very impressive, and I believe it will bring a lot of benefits to many patients, to researchers, but then in the end also will enable completely new products and insights. So Rosemary, thanks a lot for your time today and giving us the insights into Oxford Nanopore. Was really exciting. Thanks again for your time. It was a really big pleasure.
Thank you. It's been great to talk to you guys.